Hello, everyone. My name is Andrew, and I will be joined by Shail, Milin, and Salim in discussing gel electrophoresis of proteins. There are four things that we will be covering in our presentation. So we will first provide an introduction and history on gel electrophoresis, and then we will explain its purpose and the procedures and materials involved. Third, we will cover some variations to the gel electrophoresis process, and lastly, we will talk about some of its applications. So let me first begin by briefly explaining what gel electrophoresis is. So to put it simply, gel electrophoresis is a process in which macromolecules are separated based on their molecular sizes by utilizing their charges. The ionic and physical properties of various macromolecules have been studied for a long time. And so the process of gel electrophoresis has been around for some time as well. Gel electrophoresis was originally developed and used in the 1960s to separate and study nucleic acids rather than proteins. Before electrophoresis, the method for separating DNA fragments was through the use of density gradient centrifugation. This process, which was developed in the 1950s and used by Matthew Meselson and Franklin Stahl, their famous Meselson and Stahl experiment, involved the mixing of macromolecules such as DNA in cesium chloride solution. The movement of cesium ions in the solution following analytical ultracentrifugation for around 10 hours creates a density gradient, causing bands of DNA with different densities to form. However, this process is slow and costly to perform, so scientists look for different properties to utilize to separate macromolecules more efficiently. Once it was discovered that charged molecules could be separated by density by utilizing their repulsive properties to physically separate them, gel electrophoresis became a popular method for separating DNA. And while gel electrophoresis had been working fine for DNA, there are a few other obstacles that had to be overcome when applying the process to proteins. The sugar phosphate backbone of DNA is linear and has a uniform negative charge that allows DNA to easily separate within the gel. Proteins, on the other hand, greatly vary in their properties. Compared to DNA, they are larger molecules, and their secondary, tertiary, and quaternary structures cause their shapes to be nonlinear and folded in all sorts of directions. This makes it difficult for some proteins to fit in the microscopic pores of the gel, which is problematic because it would cause the protein bands to form due to properties other than density and size alone. Additionally, the R groups of amino acids can cause other complications due to their charge and ability to form bonds such as disulfide bridges with other R groups. These complications resulted in the need to treat proteins appropriately before gel electrophoresis. And in order to deal with the varying shapes of proteins, Klaus Weber and Mary Osborne tested the use of sodium dodecyl sulfate solution, or SDS, and beta-mercaptoethanol to denature proteins prior to the electrophoresis process in 1969. Beta-mercaptoethanol, also known as 2-mercaptoethanol or BME, separates disulfide bridges between amino acids. The anionic SDS solution denatured proteins so that they linearized and became surrounded in negative charge. This process would become known as sodium dodecyl sulfate polyacrylamide gel electrophoresis, abbreviated as SDS-PAGE. Since its development 50 years ago, SDS-PAGE has remained a common and practical method of protein separation still used in research today.
Hi everyone, I'm Millen and I'll be now talking about types of electrophoresis. So here's a nice little graphic that gives you an overview of the different types. So electrophoresis is broken down into capillary and zone. And from there, zone is broken down into paper or gel. And from there, gel is broken down into 4 different subcategories. Let's start with capillary electrophoresis. So this method separates ions based on their electrophoretic mobility with applied voltage. So from this, the greater the field strength, the faster the mobility, which is why it's most used because it yields faster results in high resolution separation. There are 6 different subtypes of capillary electroseparation. We have capillary zone electrophoresis, capillary gel electrophoresis, micellar electrokinetic capillary chromatography, capillary electrochromatography, capillary isoelectric focusing, which separates peptides and proteins, and capillary isotachophoresis, which is used in a discontinuous system. And then we have two different types of systems, continuous and discontinuous. And continuous system is where the background electrolyte acts as a buffer. And then in a discontinuous system, the samples are separated by different electrolytes. Next, we have zone electrophoresis, and this is when the molecules are immersed in a solution that creates a charge to mass ratio. Molecules are separated into zones or bands based on size. All components move through an agarose or polyacrylamide gel. Each nucleotide contains a phosphate group with a negative charge, and the charge to mass ratio is equal for DNA or RNA molecules. Next, we have paper electrophoresis. So this is when a filter paper strips are soaked in a buffer solution, and then a small volume of serum proteins are placed on the paper and a direct current is passed through. And we have the proteins are separated into 5 different zones: albumin, A1 minus, A2 minus, B minus, and then Y globulin. Next, we have gel electrophoresis. This is a technique to separate DNA fragments based on size. And there are many types of gel electrophoresis, specifically 4 categories. So first, we'll talk about cellulose acetate gel electrophoresis. So this is a type of zone electrophoresis that separates molecules in liquid form, such as proteins, on a membrane or a strip made of cellulose acetate. Next, we have agarose gel electrophoresis. So this method separates a mixed population of macromolecules such as DNA or proteins in a matrix of agarose gel. The proteins are separated by charge and or size, and the DNA and RNA fragments are separated by length. Next, we have polyacrylamide gel electrophoresis, and this is a powerful tool used to analyze RNA samples. When polyacrylamide gel is denatured after electrophoresis, it provides information on the sample composition of the RNA species. And next, we have SDS page. And SDS page separates proteins primarily by mass because the ionic detergent, which is SDS, denatures and binds to proteins to make them uniformly negative charged. Thus, when a current is applied, all SDS-bound proteins in a sample will migrate through the gel toward the positively charged electrode. When SDS meets with the protein, SDS's hydrocarbon tail dissolves any hydrophobic region of the protein, while the sulfate ends break down non-covalent ionic bonds. This causes the protein to lose its secondary and tertiary structure and causes it to unfold.
In this video, we will learn how to load and run DNA samples on an agarose gel. We will be using a mini subcell to perform our agarose gel electrophoresis. The subcell has an electrode wire running across the bottom of each end. This allows the electrical current to pass through and separate the DNA fragments based on weight. The first step is to align the gel so that the wells are closest to the negative side. DNA is negatively charged, and as the electricity passes through, DNA will move towards the positive or red side. Now we place the gel into the tray. Then we have to add the electrophoresis running buffer. It's important to keep filling the reservoir until you get to the max line that covers at least 2 millimeters above the gel. It's important to place your samples in the correct order according to the lanes or wells that they are assigned to be running in. When loading samples, it is important to keep the pipette tip perpendicular to the row of the wells. This helps prevent it from breaking through the bottom. It is important to make sure that you get all of your sample into the well. Otherwise, it will leak and not be able to run correctly. Once you load all the samples, try to avoid any movement or bumping of the gel, as it could knock some of your sample out of the well. When placing the lid on the mini subcell, it's important to make sure that the red goes to the red and the black goes to the black. Now it's time to attach to the power supply. When doing so, it's important to make sure that the red matches the red and the black matches the black. It's then time to go ahead and turn the power supply on. After this, we go ahead and set the correct voltage and time to run. You will begin to see bubbles start to forming. As you look closer, you'll notice that the red side has significantly less bubbles than the black side. After a couple of minutes, you will begin to see the samples moving through the gel.
Hello everyone, my name is Salim Safi and I will be presenting on the application of electrophoresis. When sample mobilities are measured at several gel concentrations, they are evaluated for the following phases. It allows for exploitation of differences in charge and shape, particle size, and the following. Samples yielding patterns with no discernible peaks are able to be evaluated by way of product size and charge distribution profiles. Charging size isomers of proteins are unable to be detected. Also, different protein conformations are able to be evaluated, and reasonable size estimates are able to be depicted. Electrophoresis has the benefit of being able to separate a much broader range of molecular sizes. Nucleic acids are able to be visualized with fluorescent staining, and extraction of bands for purification becomes more efficient. Some examples of uses of electrophoresis include separation of DNA fragments for DNA fingerprinting, which is useful in investigative crime scenes, analyzation of results of polymers chain reaction, which we know as PCR. When analyzing a specific illness, electrophoresis allows for analysis of proteins associated with that illness. In order to distinguish different species, DNA profiling is useful. Paternity testing using DNA fingerprinting is another use of electrophoresis. Electrophoresis is used to study structure and function of proteins. It is also used to analyze antibiotic resistance. This is beneficial in developing new vaccines and medications. Electrophoresis allows for evolutionary relationships to be analyzed through genetic similarities found among a population or species. Protein electrophoresis is a test that measures specific protein found in the blood. It separates protein in a blood based on their electrical charge. The protein electrophoresis test is used to find abnormal substances known as M proteins. Protein electrophoresis is utilized to detect early signs of cancer, as well as thyroid problems, diabetes, anemia, liver diseases, poor nutrition, and certain autoimmune diseases. The purpose of protein electrophoresis is to diagnose the following symptoms: unexpected weight loss, bone pain, severe tiredness or fatigue, constipation, abnormal thirst, frequent urination, frequent illnesses or fevers, bones that fracture easily, back pain, and high levels of calcium in the blood. The purpose to focus on with protein electrophoresis is examination of proteins, which are important building blocks to all cells and tissues in the body. Having knowledge of which proteins are present, absent, or a decreased amount in body fluids can be beneficial in diagnosis of various diseases and conditions. Body fluids such as blood have many different proteins that are playing important roles in transporting nutrients, removing toxins, and controlling bodily functions. When serum electrophoresis is used, its purpose is to help diagnose and monitor lymphoma, chronic lymphocytic leukemia, or monoclonal gammopathies. Also, investigate all normal findings such as protein totals or white and red blood cell counts. Also, serum electrophoresis is used to evaluate someone for inflammatory conditions such as a kidney or liver disorder. Urine protein electrophoresis is used to investigate multiple myeloma. Also, is used to investigate abnormally high amounts of protein. Cerebrospinal fluid protein electrophoresis is used to look for the banding pattern, which can be related to multiple sclerosis, to evaluate a person with headaches and look for proteins suggestive of inflammation or infection that will provide reason for these symptoms. So overall, protein electrophoresis is a useful laboratory technique for investigating protein status in the body. This allows for diagnosis of various diseases and conditions which are essential for one's health. This technique has been proven to be successful for many diagnoses and helping people with their situations. I want to thank you for listening to our presentation, and I hope you enjoyed.